everyone, we are Leonard and Judith, and this web lecture is about single-strand conformation polymorphism. Strand conformation polymorphism is a technique used to detect mutations and genotyping. The principle is based on the fact that single-stranded DNA has a defined conformation. DNA strands with a mutation will take on a different conformation than wild-type DNA. These strands will migrate differently through a gel. First of all, we need to isolate the DNA from the object of interest, which can be a virus, tumor, or genomic DNA of any organism. First, we rip the cell open and isolate the DNA out of all the cell debris. The isolation can be performed on different ways. After isolating the DNA, we will perform a PCR reaction. The targeted DNA is amplified during PCR. To target the exact region of interest, we will use sequence-specific primers. Each of the primers has a different color or fluorescent label. This is so you can recognize each strand during the analysis of the products. The amplicons of the PCR will be denaturated by heating. The single strands will be rapidly cooled to maximize self-annealing. Rapid cooling favors self-annealing because there is too little time for the strands to anneal to their complementary strand. By self-annealing, the strands will take on their unique conformation. The self-annealed and therefore folded DNA strands will be detected of mobility differences by electrophoresis under non-denaturating conditions. Because of the different conformations that the strands have taken, they will migrate differently in the gel. To detect the differences between the products, we will use a reference. The reference consists of the well type. The well type should migrate through the gel as expected. Next to the references, we will load the samples on the gel. If one of the samples will migrate differently in contrast to the reference samples, then this sample must contain a polymorphism. As you can see, sample 1 has a polymorphism in a red strand. Samples 2 and 3 have a polymorphism in a green strand. There are different applications for SSCP. It is used to detect differences in virus strains, detecting mutations in tumor cells compared to the normal healthy cells, and to detect mutations based on heritage. Nowadays, this technique is used less and less due to next generation sequencing.